Hello everyone, my name is Levi and I will be presenting our research with James and Jack, who you'll see shortly. So as you can see from the title of our presentation, we did our research on the differential effects of various sugars on the development of non-alcoholic fatty liver disease in obese mice. So why did we choose to do our research on this topic? As you probably know, obesity and type 2 diabetes have become quite a health concern in the United States and other developed nations around the world. Another illness that has been on the rise is called non-alcoholic fatty liver disease, which is, unsurprisingly, a fatty liver that is not caused by alcohol. These diseases have often been attributed to high sugar diets, particularly fructose, which can be seen here forming a sucrose dimer with a molecule of glucose. So the purpose of our research was to investigate the effects high sugar diets have on obesity and non-alcoholic fatty liver disease by looking at some of the key metabolic genes involved in the pathways of glycolysis, fructolysis, and fatty acid synthesis. Our hypothesis going into the research was that high sugar diets, particularly those containing fructose, would lead to greater obesity and fatty livers. So the methods of our research. We used an obese mouse model where, depending on the year of the study, we fed the mice high glucose, fructose, or sucrose diets. These mice were given their respective sugar in a 30% sugar solution, as well as ad libitum access, or free access, to standard mouse chow. Every week, the research team measured the food intake, the water intake, and the weight gain of the mice. At 18 weeks, we ran a glucose tolerance test, and this is done to check for type 2 diabetes in the mice. At the end of the study, the livers and the retroperitoneal white adipose tissue were excised and weighed. We then extracted RNA from the livers, reverse transcribed it into cDNA, and then used it to run qPCR to determine gene expression. We also did an oil red O stain and a liver triglyceride assay, which are done to examine lipid content and triglyceride content. And James is going to talk to you about our results.
Yes, thank you, Levi. And as he had said, my name is James Donnell, and I will be going over the results for this study. The mice weights were measured every week, and after 27 weeks of 30% sugar consumption, the sucrose, glucose, and fructose mice were all heavier than their comparative control mice populations. This was calculated using a two-way ANOVA demonstrated significant difference. This is another graphical representation of our mice's weight gain results, noting the caloric intake of the mice throughout the study. The GTT, or glucose tolerance test, measures disturbances in glucose metabolism. After the injection of dextrose, blood glucose levels were measured to determine how quickly it cleared from the blood. This determined how effectively the body is at clearing excessive glucose. The sucrose mice glucose metabolism was significantly affected, indicating that they have impaired glucose tolerance. This is also evident in the glucose mouse cohort as well. However, this impairment is not displayed in the fructose cohort. Homeostatic model assessment of insulin resistance test was performed after the mice were sacrificed. Resting glucose and insulin levels were measured in the blood, and sucrose mice were found to be significantly higher. This is also evident in the glucose cohort as well. This shows that the mice were pre-diabetic. However, there was no significant difference found between the control and the fructose mice population, which shows that they were not pre-diabetic like the other two cohorts. On the left, we can observe the oil red O stain, which tests for fat globule accumulation on the liver. Fat droplet cells are stained as red, and the blue dots display the nuclei of the cells. As we can see, there's a significant increase in fat droplet accumulation in all three of the sugar cohorts in comparison to the control. However, the glucose and sucrose cohorts contain larger clusters of accumulated fat droplets in comparison to the fructose cohort. On the right, we can see a graphical analysis of the liver triglyceride accumulation in the mice. This was done through a liver triglyceride assay. There was a significant increase in the liver triglycerides in all three of the sugar populations in comparison to their control. Through real-time qPCR, we were able to test for gene expression in the sacrificed mice to analyze how their sugar diets affected significant genes in glycolysis, fructolysis, and fatty acid synthesis. We will be observing the differences in gene expression in important rate-limiting steps in each of these processes. First, we can observe that glucokinase is significantly upregulated in both the sucrose and glucose mice. However, there is no significant difference between the fructose mice and the control. Next, we see that fructokinase is not significantly different between any of the mouse cohorts and their prospective controls. However, there is a significant increase between the FTK gene expression in the glucose mice in comparison to the fructose mice. Finally, we found a significant upregulation in acetyl-CoA carboxylase in the sucrose mice in comparison to the control. However, there is no significant upregulation in any of the other mouse populations. Now we will transition over to Jack and he will conclude this presentation. This is James Donnell signing off.
Hello everybody, Jack Farley here to finish up this presentation. At the time of the sacrifice, retinoparetinol white adipose tissue pads were collected, weighed, and compared to total body mass as an indication of adiposity. There was a significant increase in the adiposity of the high sugar diet mice, which indicates that chronic sugar consumption leads to increased fat storage. Based off our data, we know that a high sugar diet leads to things like weight gain, glucose intolerance, specifically in sucrose and glucose mice, and increased liver adiposity. Based off our research, all high sugar diets lead to non-alcoholic fatty liver disease, but glucose and sucrose seem to be more detrimental than fructose. This is in contrast to what the media seems to be saying. Fructose is still detrimental, especially when consumed in conjunction with the high glucose diet, just not as detrimental as the high sucrose or high glucose. Interestingly, although the mice on the high sucrose and high glucose diets had very similar physiological effects, they did not have the same effects when it came to gene expression. For our next steps, we are currently finishing up our last male mice model and we'll begin looking at that data soon. We'd also like to run a confirmatory Western blot in order to support our results. Mass spectrometry for the transcription factors and qPCR need to be done on liver inflammation. Speaking of qPCR, we're just starting to run it for the fatty acid synthesis gene expression in order to find more rate-limiting enzymes. To top it all off, we're beginning our final steps into publishing our research, and so that'll be what we're looking at in the next coming months. We'd just like to offer a special thanks to the N.J. Murdoch Charitable Trust, OHSU, and of course our instructor, Dr. Sarah Comstock. Without her expert guidance and amazing life lessons, none of this would be possible. Thank you, Mom Stock. These are our references.
Any questions or concerns can be emailed to sarahcomstock@corbin.edu or can be directed to our project manager, Appa. And we just want to say thank you for joining in, and we hope you enjoyed this presentation.